All right, welcome in everybody. Welcome to our Lunch and Learn on beech leaf disease with Andrew Loyd. My name is Reed Manderfield and I'm the education intern at Trees Atlanta. And for those of you who might be at your first Trees Atlanta event, our mission is to protect and improve Atlanta's urban forest by planting, conserving, and educating. And once again, welcome. This is going to be an interactive virtual learning event. So we encourage you to ask as many questions as you can think of. You can use the Q&A button in the bottom of the Zoom window, or you can use the chat to just submit them there. And we're going to be answering and discussing everything at the end. After the webinar ends, you're going to see a survey pop up. We'd really appreciate it if you would fill that out. It helps give us feedback on how we can improve your experience with us. And if you aren't already, we encourage you to sign up for our newsletter to stay up to date with all of our events, opportunities, and volunteering programs. Also, you can follow us on our social media platforms. We have Facebook, Instagram, and Twitter, and we're also working on building up our Pinterest board, so you can follow us there for more things. Coming up on December 10th, we have a fall foliage watercolor workshop with artist Patricia. She'll be teaching us the basics of beech and tulip poplar trees and floral watercolor. There's a $35 fee for materials, but we do offer a scholarship to cover that fee. I think we only have a couple spots left, so if you're interested in joining that, try and sign up as soon as possible. And next month, we are going to be moving headquarters. We're currently located in Reynoldstown. We're going to be moving to Oakland City, and our new address is going to be 825 Warner Street, Atlanta. And this is just a sketch of what our building is going to look like. We're super excited, and there's a couple of volunteer dates that we have for planting at the new building because we want to cover it with as many trees as we can. And with that, I will turn it over to Andrew Loyd. He is the plant pathologist at Bartlett Tree Experts, and he is here with us today to share about the beech leaf disease. So thank you, Andrew.
Thank you, Reed. Thank you for having me. The next presentation you guys have with painting sounds nice.
Thank you. Yes.
We won't be painting today, but maybe you guys will paint some of your beech with beech leaf disease
yes.
when that comes up. So yeah, I'm Andrew Loyd, and I work with Bartlett Tree Experts in our research division. Was formerly based out of our lab in Charlotte, North Carolina, but recently moved this year to support our Southwest operations. And I live in the Fort Worth, Texas area and work at out of the Botanic Research Institute of Texas in Fort Worth. But today I'm going to be talking about a bit of the developing story. Isn't everything today, but we're going to talk about beech leaf disease, and I gave this clever title of Microscopic Worms Causing Macroscopic Devastation. So we're going to talk about beech leaf disease and what it is, where it started, and what's causing it, and then really get into some of the research that I've done focusing in on managing this disease. So a few other people to thank, and we're part of this mini projects, Matt Borden at our lab, and Caitlin Littlejohn and Amber Stiller. So I think that this quote kind of describes, the development of an emerging problem. And this is akin to the last few years as we've all lived in the pandemic world. When something new happens, this Kurt Vonnegut quote kind of sums it up. We have to continually be jumping off cliffs and developing our wings on the way down. And that's kind of been what how this the foundation of how we've been looking at this disease is. It continues to spread and cause problems on beech, and we're learning every day something new about it. And it's the learning about the biology and the ecology of the problem. We can use that to come up with novel management strategies. So beech leaf disease, the story begins in northeastern Ohio in Lake County, just northeast of Cleveland, where people started to notice some weird symptoms on beech trees about 2012. So these symptoms would appear like this. So the beech tree would have leaves that would come out, and they would have this striping pattern between the veins, where you would have a discoloration or a darkening between the veins of the leaves when they would emerge. And they also started to notice that after several years of seeing these symptoms on an individual tree, the leaves would actually come out and look somewhat crinkled or malformed, as you can see in this picture here, right here in this section. Again, showing you this very conspicuous symptom that exists. It's really easy to see through these paper thin leaves of beech if you look at them through to the sunlight. This disease not just affects American beech, we started seeing it on planted, cultivated beech in landscapes. And here it is on a European beech cultivar, the fern leaf beech or split leaf beech. And you can see that darkening pattern on these leaves as well. And here's a columnar hedge cultivar of European beech, and you can see this discoloration in between the veins, and in this case, causing them to be somewhat yellow. And here's another photo of showing that crinkled or malformed version of the leaf after several years of being infected with the disease. And here's another shot of it on European beech. And so you can imagine that with that darkening, what's going on there, especially in what's going on in the crinkled state, it doesn't take a rocket scientist to think that probably photosynthesis is going to be affected. And so when you have a large portion of the canopy infected and having these symptoms, you're really going to reduce the photosynthetic machinery of the plant, which the plant's not going to be able to produce its photosynthates to grow and survive. So over time, you can what happens is that we see buds actually fail to survive and they desiccate, and then you get thin canopies like you see in this picture of an American beech. And over time, you can see on the left an American beech and a natural planting that has extensive dieback, and that's really from the death of these buds, and a European beech on the right that has lost 90% of its canopy from this disease. Now, this didn't happen overnight. This was several years of infestation, and so the plant didn't produce photosynthetes to overwinter, we suspect, and so it had no carbohydrate storage to form new leaves the following season, and this is what you were left with. So there's been a lot of mass media and press on this because beech is a very important species in the eastern ecosystem. It's also a very prized ornamental landscape plant where people plant it as a shade tree. So here's an article. Here's another one. Mysterious disease is killing beech trees. You can see the date here, 2019. People weren't sure of what was causing this. They knew it was spreading. They knew it was affecting a lot of beech, but still there was kind of a debate on what was going on. To speak to the importance of beech, this is just a U.S. Forest Service distribution map showing that beech, can be found all the way into northeastern Canada and down all the way into the panhandle of Florida and as west as East Texas. So this species is certainly a large part of our eastern forest, especially in the Appalachian mountain range, where beech maple forests are a late succession hardwood forest that we see. And they're certainly providing a lot of ecosystem services as well as wildlife, because beech produce these fruits, these beech nuts, and these are a source of nutrition for migratory birds, resident birds, deer, other wildlife, bear. And so, this tree is not just a tree and not just providing food source, but beech trees and late succession forests will also provide canopy cover for understory plants. And so this disease can have a real impact on the forest and not just beech trees is what I'm trying to get at. There's a domino effect that when you disrupt the ecosystem, something else is going to have to come in and take the place of the beech. A lot of the understory things are probably going to suffer. wildlife is going to suffer. And it's likely this day that we live in that potentially invasive plant species will move in to replace the beech, and we'll have a less healthy forest system. So this brings up to this idea of, what's moving this thing around. I've even told you what causes the disease yet. But because the beech trees are such an important source for nutrition for birds, the thought came about that maybe birds are helping move this pathogen around that's causing the disease. And there's some work going on with the Forest Service and the Holden Arboretum, where they're actually trapping birds and then looking for the pathogen, which happens to be a nematode, spoiler alert, in the fecal droppings of the birds as well as on the bird feathers. No birds were harmed in their studies. So that was kind of jumping ahead of myself a little bit, but this is where we're at now. So again, remember, this disease was first found up here in Lake County, Ohio, and each color represents a different year of detection. And you can see this disease has spread pretty widely to the east of its original detection in north, but it's also starting to spread westerly and north into Michigan and Western Ohio, but it's also found in Ontario province, Canada. As far south as a isolated population in Northern Virginia. So this thing in the last 10 years has spread quite rapidly, and it's quite concerning. So again, remember, the first detection and the first observation of seeing these symptoms was in and around 2012. And it wasn't until 2018 that people started to figure out maybe what's causing this thing. And so in Japan, there was a paper that came out in 2018 that described a new foliar feeding nematode that was causing a symptom very similar to what we were seeing on American beech in the US, causing these leaf discoloration between the veins, kind of gall-like tissue. And these authors report, this was on Fagus crenata in its native range in Japan, and they suggested that this thing was just an anomaly. It wasn't causing a whole lot of plant health concerns, problems for these beech trees. It had just been reported, and they didn't really know the cause. And it was reported as being observed in and around 1960. And so in 2018, they described this nematode Littlelinchus crenatae as the pathogen that was causing this symptom in its native range. And in and around the same time, there was a plant pathologist in Ohio, Dave McCann, that extracted a bunch of nematodes from the symptomatic American beech trees in Ohio. And so these were sent off to the USDA Beltsville Nematology and Mycology Lab to Dr. Lynn Carta, who then was able to describe this nematode as a subspecies of the one that was described prior to in Japan. So Littlelinchus crenatae McCannii is the foliar nematode that is causing beech leaf disease in North America. Now, this is likely that this nematode was introduced some sort of way from Asia into the United States, probably in Ohio first, and it's continued to spread. We don't really know this for sure yet, but it all makes sense from a invasive pathogen story. So typically when we have invasive pests or diseases, they cause a lot of problems in their naive stand. So if you have a pest that's introduced from Asia to the United States, it has more potential to cause a lot of damage and harm to the native trees here because they have not co-evolved together. So a perfect example of this is emerald ash borer on ash trees, where the insect pest does not cause any problems on Asian species of ash. In the United States, it wipes out all of our native ash. This story of beech leaf disease fits that pretty well. In Japan, Fagus crenata is not really being affected from a health standpoint. These nematodes exist there. Here, they're causing a lot of problems. So I think that it fits, and we'll learn more as we go. So this is a microscopic photo of our nematode, the pathogen that causes beech leaf disease, Littlencus crenata McCannii. So in plant pathology world, to determine if something does cause a disease or is associated with the symptom, we have to take that organism out and try to recreate the symptom. And this is called Koch's postulates. So David Burke and others at the Holden Arboretum did this very thing. So they extracted nematodes from symptomatic leaves. They inoculated buds with eggs and juveniles and adults of this nematode. And then lo and behold, they got the same symptoms develop on those leaves that came out of the bud. So it's pretty well determined that this nematode is the causal agent. And from someone that has extracted, hundreds of samples of symptomatic beech leaves with this disease, you'll learn very quickly that, one tiny band of a leaf can have thousands of nematodes in it. It makes sense that this is our pathogen. So here's a photograph of a bud, and you can see this silver looking thread thing. This is our nematode. So nematodes are microscopic worms, and these nematodes will overwinter in the buds. And as those leaves plump up in the spring, those primordial leaves are fed upon by these nematodes, and the nematodes will lay eggs and they will continue to feed. And when the leaves come out, they have their symptoms like this. So there was some good research from our Canadian partners in their forest ministry. Sharon Reed led this team where they were tracking when these nematodes were most active in their mobile state over the course of a growing season in Ohio and in Canada. And so they collected leaf samples, they extracted nematodes, and they looked at, how many nematodes would come out of these symptomatic leaves over the course of these months. So you can see that there's not a whole lot of activity. Beech full leaf expansion in Georgia is probably going to be sometime in late April or May. Up further north, it'll probably be June. But you can see that there's really not a whole lot of mobile nematodes until about July, and then they start to peak up in August and finally peak out in September. So this is an interesting find because this tells us that these nematodes become active in this state. The damage has already been done in the bud, and where do they go? So in and around this same time, you have a few things going on. First, you have buds forming for next year's leaf set. So beech are a determinate species, meaning that they produce one set of buds for the canopy for that season. They typically don't have a second flush of growth in a growing season unless they are sheared as a hedge or pruned hard. They will form that one set of buds. So these buds are forming in and around this time these nematodes become active. So where are they going? They're leaving the leaves and they're entering the buds underneath the bud scales. The other thing that's happening in and around this time that these nematodes are most mobile is these beech trees are producing their nuts. So this food supply for wildlife and birds and other things. And this really speaks to maybe how this thing can spread around and why it's moved in the last decade from Northeast Ohio, as far as Maine and Virginia. So what happens with the leaves? I told you that it disrupts photosynthesis. Well, this is some really cool scanning electron micrographs of a beech leaf. And what you're looking at in this top picture, where my laser pointer is, this is the cuticle of the leaf. And then down here, this is the underside of the leaf. So these are all mesophyll cells that help, have chlorophyll and do photosynthesis. And you can see this is what a healthy leaf looks like. Well, when the nematodes have infected a beech leaf, this is what those mesophyll cells. So you can understand that photosynthesis is really going to be disrupted. And you can actually see here's a worm, a nematode right here. Here's another piece of a nematode here. So this is quite damaging to the plant. And okay, you might have one band on a leaf that might not be so harmful to the plant, but when you have 100% of the leaves that have 50 to 60% of the leaf area damaged like this, you really reduce the photosynthetic capacity of that plant. So how we think this disease works is very akin to a story that I read to my children, Shel Silverstein's The Giving Tree. So these nematodes come and they take from the plant. The plant has no more carbohydrates, no storage left. It can't give anymore. And that is how this disease works, we think. And it's a carbohydrate depletion disease. So once the bank dries up with storage reserves, the tree it fails to produce healthy buds and leaves, and it dwindles out. So it's a progressive disease that doesn't kill trees quite quickly, but over time it affects them and they become stressed and they go into a state of decline. So here's a forest picture on the right that's been had the disease for probably at least a decade. And this forest, once in the summertime when this picture was taken, it was all shaded. It was very dark. You couldn't see a whole lot of sunlight coming through. And now it's starting to open up because these beech are getting thin. We've seen trees die within 5 years of first symptom onset. In other locations, we've seen them die quicker. But one thing that we do know is that the smaller the tree, the quicker it dies. And so we've seen saplings and regeneration in a forest suffer the most and the quickest. And it makes sense based on that carbohydrate depletion hypothesis that smaller trees are going to have less storage reserves. And so this disease is going to be more damaging on them compared to larger trees, which might take longer to deplete that carbohydrate storage. And one of the cool things that we found by doing a study with a graduate student at the Holden Arboretum in Case Western University and David Burke was we started to think about beech a little more holistically. We know this is a foliar disease caused by this foliar feeding nematode, but we also know that beech are ectomycorrhizae forming trees. So ectomycorrhizae are a symbiotic relationship between a fungus and tree roots. And the fungus helps the plant by mining for phosphorus and other mineral nutrition as well as water. And it makes the surface area of that root mass significantly larger. And in return, the plant produces photosynthates that then get sent down to these fungi for nutrition. So it's kind of a partnership between these two. And so we thought, well, what happens to these ectomycorrhizae when you have beech leaf disease? So trees in an orchard setting were sampled, whether they were characterized as good or healthy, mild symptoms, if any, moderate or intermediate, and then bad. And then root tips were counted based on having ectomycorrhizae. And so different morphological characters can tell you if the root tip has been colonized by a fungus. So these are swollen root tips, and this is typical of a particular species of fungus. But this is what we would look for under the microscope, and we would count how many of these root tips have an ectomycorrhizae relationship. And what we found was that the healthy trees had, on average, a much higher incidence of roots being colonized and having this relationship, where it started to go down when you got into the moderate disease class, where 30–40% of the canopy is affected. And then finally, the 100% of the canopy being affected by this disease, you had significantly lower percentages of those root tips that had ectomycorrhizae relationships. So again, this speaks to this idea that the above ground disease is depleting carbohydrate storage because it's not photosynthesizing as well. And it's also disrupting the below ground ecology of this symbiotic phenomenon, mycorrhizae. And so this is somewhat troubling. So I like to think of it as this, the mycorrhizae aren't getting their fair share. So they go away, and you might get replacements with weaker fungal partners, or you might not have them at all, and the beech can suffer. And this is not only going to affect that individual tree, but it's definitely going to affect the regeneration of beech stands. So that's kind of the nutshell of this disease, where it is, what's causing it, how it works. And now I'm going to jump into some of the research that I've been doing on management. Can we manage beech leaf disease with pesticides or products, or is this something that's just not going to be feasible, and we will lose the American beech in our plantings and forests and such. So I think from this standpoint, to start, we have to consider, what are we trying to manage? There's a difference between managing beech and natural stands in a forest. And pesticides probably aren't going to be the dominant answer for this, because there's too many trees, there's too much of an environmental impact potential, and it's going to be expensive and very challenging. So there are groups of people, in particular, I know of a group in New York State that is looking at some silvicultural practices, like removing diseased trees and then selectively removing to open up canopies and thin them to reduce the spread and likelihood that you'll have a quick disease progression in a forest. Now, for high value specimen trees or plantings in collections or arboreta or landscape plants, I think pesticides could be a big part of management if we find something that could work. So we're really going to be focusing in on that to set your minds to this foundation. So where do we start? Nematodes, foliar feeding nematodes on woody plants causing diseases is really an anomaly. Okay, there's very few of these reported about, and so therefore they really haven't been studied a whole lot. There's a boll infesting nematode pest that affects exotic pines planted in this country. Pine wilt disease, you may have heard of, caused by the pine wilt nematode, is probably the closest thing we have in trees as far as a above ground nematode pest. That one is obviously a bit different, but it has been studied. So maybe that's where we start. So what we did is we put together a list of active ingredients of chemistries that have nematicidal properties, but maybe they're not registered for nematodes. They've been shown to be effective in other systems with these active ingredients, such as azadirachtin, acephate, fluopyram, abamectin, but they're not necessarily labeled for nematode control with the products that exist in the landscape. Now, on the other hand, there are products that exist that are nematicides in agriculture or turf grass systems where people have had good success with some of these active ingredients. And then finally, we meet in the middle where we have one class of chemistry, the avermectin class, including imamectin benzoates. This would be like triage G4 or mectinite or abamectin that are labeled for pine wilt disease. And there's really good research to show that it's effective against that disease. So this is kind of where we start. Maybe we look at these active ingredients and try to figure out which ones work and which ones don't. So as I mentioned, this pine wilt disease, this is a disease in this country caused by a native nematode, and it doesn't affect our native pine species. It only affects exotic pines. So Japanese black pine would be a common one that you would find planted in Georgia. Austrian pine you'll find occasionally. Anything that's not native to this country can be affected, and it would be a devastating disease. And just as an aside, this nematode has been introduced into Asia and is causing widespread damage in native pine forests in Korea and in Japan. So the research that existed looking at imamectin benzoate, so this would be your mectinite or triage, they injected trees with imamectin benzoate, or they did not. They had paired treatments. And then they inoculated these trees with nematodes, the Bursaphilincus xylophilus, and they looked at mortality over a period of 3 years. You can see that every other line is treated and paired with a non-treated. So you can see 100% mortality in the non-treated, and the majority of the ones that were treated have 0 mortality up to 3 years after. So this chemistry seems to be quite effective against this particular nematode. So would it be effective against beech leaf disease? So we wanted to start out in the lab, just looking at this chemistry to see if it would be effective, and then following up with field trials. So in vitro, we took samples that were infected, we extracted nematodes from them, and then we treated them with different concentrations of imamectin benzoate, so 43,000 parts per million down to 43 parts per million. And we had a water control. And then we counted nematodes throughout a period of 24 hours for mortality. And you can see that all of the treated ones with imamectin benzoate resulted with 95 to 100% mortality. Whereas our water control, they were pretty much all still alive and wiggling in the Petri dishes. So we found out that this chemistry seems to be effective at this nematode in the lab. So we took it to the field and we injected trees using a system that looks something like this with a macro injection type system. We diluted the product quite a bit because imamectin benzoate is quite viscous and we needed it to move into the leaves and the buds. And so to do that, we injected the trees with 10 to 20 mils of imamectin benzoate product at a 4% product rate. And we diluted that in 250 mils per inch of diameter. We did this in a bunch of 3 locations, and we had non-treated controls to check, and we looked at the nematode numbers and the treated versus non-treated trees, and we did pre and post canopy ratings. So what did we find? So when we look at the nematodes in the dormant buds, so they overwinter in the bud sheaths, we found no difference, statistical difference in the number of nematodes per treatment. So that's not looking promising. When we rated the trees initially, the gray bars are showing the percent canopy with beech leaf disease symptoms. So you can see they were around 25 to 30% of the canopy had symptoms in 2021 when we did our treatments. But by 2022, these trees did not recover. They in fact got worse in all treatment groups. So imamectin benzoate may be effective in the lab, but it's not getting to the leaf tissue at the concentrations it needs to suppress the nematode population. So then we wanted to look at another product, Acephate. This is an older chemistry insecticide that has been shown to have some efficacy on some nematode pests. Here it's showing on root knot nematode of peppers, having a 81 to 76% reduction in nematodes on pepper plants. So we figured we would try it because there's a landscape label for Acephate. So we had trials where we injected with the soil rate, high rate on the Lepitech label, that's the trade name of the product. And we did this in the late summer or at bud swell, and we had compared non-treated controls. We did this at 2 different locations and both on a mix of American and European beech. We did the same sort of rating scheme, counting nematodes as well as looking at symptom development. And what do we find? Well, we found that there was no difference again in the number of nematodes across the treatments compared to the controls. So this chemistry doesn't really seem to be effective, and this was mirrored by the disease severity ratings pre and post treatment. So the plants did not improve, they got worse, and so we saw no efficacy from Acephate as an active ingredient. So then we go back to the drawing board and we look at the other active ingredients that we have come across. And here is an example of some efficacy on root knot and lesion nematodes on turfgrass putting greens with an active ingredient called fluopyram. So fluopyram is a fungicide that happens to have nematicidal properties. And you can see a core from a putting green grass core and the root system that developed on it when it was treated versus a non-treated part of the putting green. So this active ingredient looks pretty promising for as a nematicide. The other one is this abamectin group. You can see that the quality of the turf grass in these photos compared to the non-treated, it is much improved whether it was treated with fluopyram or abamectin. And this was work from Billy Crow's group at University of Florida. So how do these actives work on our nematode that's causing beech leaf disease? So we wanted to start out in the lab again, and what we did was we had cuttings of symptomatic leaves, and we took hole punches from those symptomatic leaves in the late summer, and we extracted nematodes from these as a pre-count. And then we took the cuttings and we made a fresh cut. These were overnighted to us as if they were propagation material. We stuck them into tubes that had these different products, and we had a water control. So we had Indemnify, which is a fluopyram product. We again tried our imamectin benzoate And then we tried this product called Contos that is spirotetramat, which is a known miticide that also has some nematicidal properties. We allowed these cuttings to sit in these solutions at different concentrations for 48 hours under fluorescent light to allow them to uptake the product. And then after 48 hours, we extracted nematodes again. And then we compared pre and post count extractions. And so what did we find? Well, what we found with the control, the ones that were sitting in water, there really was no difference in the number of nematodes we extracted pre and post, which is good. That tells us our experiment, worked pretty much the way we needed it to. But what was statistically significant were the fluopyram treated cuttings. And so there was a significant reduction in nematodes that were extracted post-treatment with these chemistries. So this was promising for us. So we wanted to take this to the field. So we used this turf grass product, Indemnify, the active ingredient fluopyram. At a nursery, we did foliar applications with this product. We also did foliar applications with an abamectin product. And then we had non-treated controls. We had about 8 replicates per treatment at a private nursery in Perry, Ohio. And these were mostly European beech, a variety of cultivars. We rated them the same way we've done the previous experiments. And lo and behold, we found something that finally is going to work. So what we found was, compared to the control, the dormant buds that of the trees that were treated with Indemnify, the fluopyram product, had significantly lower numbers of nematodes than both the control and the lucid in oil or the abamectin treatment. So that was great. Well, how did this reflect in the foliar ratings? So what we saw, both lucid in oil and the controls, the disease symptoms went from around 30% of the canopy having symptoms to about 50% the following year. But that was not the same story for our fluopyram treated trees, where we saw a reduction of by 40% of the canopy. So pre-treatment, these trees had about 50% of the canopy with symptoms. Post-treatment the next year, we were able to reduce the disease to roughly around 5 to 10% of the canopy with symptoms. So this was great news. So what is this fluopyram? This is directly from Bayer, now InView, its website. This basically, in a nutshell, fluopyram is a fungicide that happens to work on nematodes, and it basically blocks their energy producing pathways. So once they're exposed to this chemistry, they can't produce energy anymore, and so they basically fade out and will not reproduce, will slow feeding and activity. So this is kind of how it works. What we found out was that Bayer also makes a product that has a landscape label. It's labeled to be used on shade trees like beech that has fluopyram. It's called Broadform. So because of our positive results with the nematicide Indemnify, we decided to start looking at this one that's already legal and labeled to use on trees. So we have 6 different trial sites ongoing. We did applications with Broadform at the high labeled rate 3 times on these trees, and we had non-treated control to compare. And so we're very early in the data sets, but what we can tell you is that we've gotten nematode extractions done from leaves following 1 application. And what we found is, compared to the non-treated controls, there was a significant reduction in live nematodes after 1 application of Broadform. And then after 2 applications of Broadform, we saw just the same trend. We saw a reduction in the number of nematodes. So again, we're seeing the same promising result with this product. So this is good news for beech trees. I didn't put it in the slide set, but we also recently did some bioassays where we took cuttings, we sprayed them with different concentrations of Broadform, and we saw a very similar thing, that after 5 days post-treatment on these cuttings, it was very difficult to find live nematodes in these leaf tissues compared to the controls, which were treated with water, where we can continue to extract nematodes out of the plants up to 2 weeks after collecting the sample if kept hydrated. So there's obviously some pros and cons to this. I mean, the pros is pretty easy to know that it works on the nematode. That's great. But making foliar applications to large beech trees can be difficult, and you really need to manage drift, and we don't want to have problems with drift. So something like a root flare injection is great because you can put it directly into the plant and it gets to where it needs if it's efficacious and it works. But right now, all we have is this foliar option. So it's something that we're mindful of, but it works, so we can reduce this disease. So that's the benefit of it. Just wrapping it up from the management side, we definitely have tried some things that haven't worked and moved on from them. But I'm happy to say we found something that did work. We have other trials ongoing with some injectable or soil applied products. So thiabendazole is a fungicide called trade name Arbotect. We use this for Dutch elm disease management, but thiabendazole is an old school nematicide as well. And it was used in veterinarian medicine back in the 50s and 60s to control different gut nematode diseases of cattle. And it's also been used in human medicine. So just thinking about this chemistry, could it work for this nematode? We have an injectable product available to us. We have a trial underway. We also have a trial underway with a growth regulator called Cambistat. And so Cambistat is a product that it inhibits gibberellin synthesis. And gibberellin is a plant hormone that is involved in cell elongation. So the thought here is that if you apply Cambistat to a beech tree, cell elongation is much smaller. So you'll have smaller leaves. It'll have a higher density of cells per area, and the leaf cuticle is quite thicker. So can we disrupt the anatomy of the plant so much that the nematodes have a hard time feeding on the tissue, or they have a hard time leaving the leaf tissue that's diseased and moving into the buds? So that's kind of the thought there. And we have that will be compared to a non-treated control. So more to come on this. We're constantly looking for other options, but we do have something that works with fluopyran. So, this is the research team where, have been searching for the Holy Grail, but we have found something that is successful. And so we're moving towards looking at it from a more integrated pest management program on how many applications are necessary, at what time is it most optimal to make these applications, and maybe we can make a difference in saving the beech trees and not having them go the way of the American ash species or the chestnut or the elms. So that is our hope. Still have a lot to learn, and I'm happy to entertain any questions after I give thanks to all these institutions that have helped us, as well as these individuals that were key in a lot of the work that we have done on this disease. So thank you so much, and I will take any questions.
Thanks, Andrew. That was wonderful. I especially love the last couple slides there with the Holy Grail and Star Wars.
Absolutely. So any questions in the chat or?
Not quite yet.
Okay.
Okay, we did just get one right as I said that. So Alex said, intriguing topic and something I'm not sure enough of the public has much, if any, knowledge about. While typically used in treating for boring insects, since it's highly toxic to invertebrates, have imidacloprid or other neonicotinoids been explored for use in systemic treatment? And sorry if I butchered those names.
No, you did great. Yeah, it's a great question, Alex. We have not tried those. There is some evidence in other systems that neonicotinoid insecticides have maybe low levels of efficacy against nematodes, root feeding nematodes, but we have not tried it. We have trees in the field that have the disease that have been treated that we've looked at, and it doesn't seem to have an impact, but it not saying that it's impossible that it wouldn't work. It is something worthy of exploration. We just haven't done it yet.
And Kendria said, you may have mentioned this already, but are these species of nematodes foreign?
Yeah, Kendra, we suspect as much. It fits an invasive species kind of profile in that they're spreading rapidly from a concentration area and moving outward, and they're causing a lot of problems. Whereas this nematode in its native range, the species Litholinchus crenata in Japan, doesn't cause real big issues on Fagus crenata, the Japanese beech. So we suspect there's a lack of co-evolution and co-adaptation, and so we're seeing more problems. And from that, we suspect that it's introduced.
And Taryn asked, is there any research on what is controlling the nematode population naturally in Japan?
That's a great question, and something that is certainly intriguing. I don't think so, as far as I know. That you got to recall that nematode in Japan was described in and around the same time that we identified it as a causal agent here in the United States. So there's been limited work that has followed up on that in the native population. But it's certainly something that would be something to look at, because biological control would be a fantastic option for this disease. Now that does take a lot of effort. Classical biological control would be going to the point of origin, finding some maybe intimate pathogenic fungi or other things that could feed on the nematode. But if we find that, it still has to go through the same hurdles of a pesticide at the EPA level to make sure that fungus or other microbe isn't going to get out and cause problems on our other natural native nematodes, let's say. And then how do we deploy it in the tree? So I think it's something that we need to look at, but I think there's going to be some serious challenges with getting something like that to work. But we have had conversations with some Korean scientists that do have a biological control for pine wilt nematode. And it's a fungus that they inoculate into pine trees, and it can cause curative and therapeutic benefits to these pine trees from that standpoint of that disease. So it's not unheard of that there may be something out there, but we're quite a bit of ways away from that.
And then William asks, are there any projections for spread in the next 10 years?
There haven't been any modeling papers that have looked at that. But what I can tell you, William, is that in the last 10 years, we have found this thing into 13 additional states, a Canadian province, and it seems to be spreading quickly. So I would suspect, that population in Virginia was detected last year in 2021. I think it's going to radiate out, and eventually we're going to start to see it further south. But as far as predicting it, I'm unsure.
We've got another question here from Alex. Have you any insights about why this and most other invasive tree diseases in the eastern US seem to get a foothold in more northerly states, then migrate down the spine of the Appalachians until they either wipe out of reach and equilibrium with trees in southern forests?
Yeah, I think a lot of it has to do probably with ports of entry. Now, this could change over time, and I'll get to that in a second. But if you look at emerald ash borer, it was introduced first in Michigan, and it's spread since. And that is one that does cause problems the further south it gets. It's into Georgia and South Carolina. It's made it down to Texas, and it's killing a lot of native stands of ash trees. So it's not one that is less damaging. And I would also say that there's another example of one that was introduced in the South, laurel wilt disease, killing red bays and sassafras trees in the southeastern United States and west Texas, was introduced near to the port of Savannah. And it was a beetle-borne fungus that causes a wilt. So there are examples of the opposite. Now, I think that what's problematic is that the Panama Canal, if you don't know, was expanded a few years ago to allow for larger cargo ships to come to the ports on the East Coast, and including Miami and others. So it's possible that we're going to start to see more invasive start in the South because of this, because before, the major shipping ports were mostly, the large majority of them would come from Asia to California. And then these would get unloaded off of ships. They would be put into 18-wheelers and moved across the country. So it's kind of a very hard lifestyle to actually get some introduction in place. But if you can take something that comes from a country that has a similar forest composition to ours, and you go straight to the source of the plants, it's going to be a lot easier for these introductions. So I think as long as we have a global economy, which might save our pocketbooks some money because of cheaper goods and services, we're going to have more invasive exotic pests and diseases introduced into this country, unfortunately.
Oh, that's really unfortunate. Yeah. And then Susan asked, she thinks that you mentioned another species that's affected by the nematode. If so, what was that species?
So the nematode is affecting European beech, Fagus sylvatica, and it's affecting American beech, Fagus grandifolia, by and large. Now, there are some botanical gardens and arboreta that have found this nematode on other beech species, but those are mostly one-offs. And so by and large, we're seeing it in natural stands of American beech, planted stands of it, as well as all European beech cultivars.
All right, well, it looks like that's all for questions. There are a lot of them, so glad everybody was interested in this topic. That was very interesting. So thank you a lot, Andrew, for joining us and sharing your experience and expertise.
Thank you, Reed. Thank you, everyone.
Thank you. And just a reminder that once the webinar ends, a survey is going to pop up, and we would really appreciate it if you guys would fill that out, and hopefully join us for more events and lunch and learns. Have a great rest of your day, everyone. Bye.
Bye-bye.